Welcome to Lecture 10 in DIY transcriptomics. Now that you have your differentially expressed genes or differentially expressed transcripts, we need to now group genes together or transcripts together based on their behavior across the samples that we've measured expression in. And that's a process I'm going to refer to as clustering, and that process of clustering results in modules of coordinately expressed genes. You'll hear module and cluster often used interchangeably. For simplicity in this lecture, I'm going to refer to clustering as the verb, and the thing that clustering produces will be our modules. Okay? All right, so I always like to take stock of where we are. You've made tremendous progress so far. We've moved all the way through from getting our data in to filtering it to working with that data using different plotting functionalities and data wrangling and tidyverse tools within R. You then learned how to access public data either on the SRA or via this ArchS4 resource. And then we came back, now that you have multiple ways of getting into this workflow with your own data or public data, we've come back now and recently actually carried out a formal differential analysis to identify differentially expressed genes between patients with this disease that we're studying in this class and patients or endemic controls. So today we're here. We're sitting here with these DEGs, and you might be thinking at this point, well, all right, so we have these DEGs already. Why isn't this class about understanding functional enrichment? What kinds of gene functions are represented in the genes that are upregulated and downregulated? And the reason we're not jumping ahead to that, that's actually the next lecture, the reason we're not doing that right now is because we have in our data set at least two sets of genes. We have genes that are higher in the skin of patients with this disease, and we have genes that are higher in healthy controls. And so it's an important thing to recognize that just because a gene's differentially expressed does not mean that it is likely to be involved in the same biological processes. In fact, we may very well expect that the biological processes perhaps enriched in the genes that are higher in disease may be very different than the ones that are enriched in the control patients in this case. And so that really sets the stage for this lecture, which is we need to separate out genes that are behaving in different ways in our set of differentially expressed genes. And the reason we do that is there is this underlying assumption in biology that genes that behave in a similar manner are more likely to be functionally related than genes that are behaving in very different ways, right? So it's a very basic idea, but it's sort of fundamental to the motivation of why we move forward with something like clustering. So our data set is really, really simple. And so it's another important thing for you to recognize. We only have one possible comparison, disease versus healthy. But the norm now is to actually have far more comparisons that are made in a single RNA-seq experiment even. You might have multiple treatments or time points, in which case you would have multiple volcano plots like this, and therefore multiple modules that could be pulled out by way of a clustering analysis. In our case, we can only have two modules, genes that are turned up and genes that are turned down. All right. So I will demonstrate everything with respect to clustering and modules using this data set, but I want you to be aware that there's a lot more value from clustering and module identification as your data set gets more complex. Okay. And I'll give some examples of that as we move through. All right. So with that in mind, then, our goals for today are to be able to interpret and construct heat maps. And I'm going to emphasize interpret here because I think even though heat maps are a common visual, particularly for RNA-seq, but really for many different kinds of genomic data sets and approaches, I'm not sure everyone always understands what they're showing when they show a heat map. And it's actually a common situation that I see postdocs, grad students, even faculty get into, where they're showing a heat map and they may not exactly understand the underlying data and how it leads to that visual representation. So we'll talk about that. We'll also have a little bit of fun talking about color choice. Is it critical? No, but I'll actually give you some examples of where color can have a big impact on who can interpret your heat maps. We'll then step away from sort of the methods we use to make heat maps and cluster our genes to identify modules, and we'll talk about other clustering methods and why they may be useful to us and when they may be useful, and I'll even demonstrate one for you. And that alternative approach to clustering is going to be via a Python software called Clust. And again, this is something I have installed on my laptop, and I'll run it on my laptop to show you that it works well. And I linked to it in the lecture page for today, as well as some example data, so that if you install Clust, you can give it a try. But it's not required to do any of the work that we're going to do in this lecture. It's sort of extended learning. I always like to give you those opportunities to learn more than what we're just doing in R. And then, of course, today is going to deal with starting and finishing the entire step 1 script, the step 6 script, sorry. Okay, so what's our goal with clustering? Well, rather than jump into our data set, which has thousands of differentially expressed genes, and try to articulate the goal in that relatively large sea of gene expression, I want to start with a simpler data set. And that's one I've just made up here that has 4 genes, A through D, and 6 conditions. And I just decided this was going to be a circadian rhythm study where these 4 genes were monitored across 3 different day-night cycles: day 1, night 1, day 2, night 2, and day 3, night 3. All right, if we just use ggplot to construct this, and if you want to see what I did to make all the plots I'm going to show you, I've provided a bit.ly link here that you can see the R script. But basically, if we were to plot that, we would see our 4 genes behave like this. We have 2 relatively lowly expressed genes that follow opposite circadian rhythms, one being high in day and low in night, and the other opposite of that. And then we have 2 other genes, C and D, that are much more highly expressed, but still follow the same circadian patterns, right? One is high in day, low in night, and the other is opposite. So our goal with clustering is to find these patterns of covariation or coordinate expression across our samples. And in this case, what we're trying to accomplish is to group genes that are behaving in a similar way together. So in this case, the genes that are behaving in a similar way are genes A and C that are low at day and highly expressed or more highly expressed at night, so they follow a night cycle of expression. And D and B, genes that start off higher in the day and whose expression is reduced in night cycles. All right, and so we want to avoid situations where we might accidentally cluster these 2 genes together simply because they're more highly expressed. So we care more about pattern of expression than we do magnitude of expression, and that's going to become important as we go through today's lecture. All right, so let's go ahead and start the step 6 script. As I said, we're going to take a step back a little later in the slides and talk in more detail about other clustering approaches. So I'm heading over to RStudio. I have my R project for the course open, and I've already downloaded the step 6 script. If you haven't done that, you're going to want to go ahead and pause the video and go get that script from the course website. All right. We're going to do the same thing we do every time we open a script in this class. We're going to go Edit, Folding, Collapse All. So here are the code chunks involved in getting through the work today. Our usual introduction and a loading of packages, which we'll talk about in a minute. We'll talk about some colors. Again, not maybe the most important bioinformatics thing you'll do, but it'll be a fun part of it. And then we'll just review exactly what data we should be coming into this script with. You've already generated this data in step 5, but I'll just give you a quick refresher on what that data is. And then we'll cluster. Again, we're going to use clustering approaches to group together coordinately expressed genes across our samples. So we're going to cluster these DEGs and then produce a static heat map. We'll then make some interactive heat maps, and I will probably skip over or just talk you through this section because I'm having some issues in R since updating to the most recent version that are causing some of these interactive heat map to not be plotted properly. So it might work for you, it might not, but it's not essential. I'll then show you as sort of an option that some of you may want to explore depending on your data, how to simplify heat maps and show only a single column for each group of animals or samples. We'll then cut out the modules of interest. In our case, again, we only are going to have 2. We'll export the data from R. And then I provided another optional code chunk if you want to just read in a set of genes and make a heat map out of them with no clustering at all. And then we, of course, have our essentials code chunk. All right. The introduction you should be familiar with. We're going to make heat maps. We're going to identify modules based on clustering. The packages that we're going to load, we're going to use the tidyverse and ggplot again. You're going to use lima, but only for one of the functions in this optional code chunk for how to simplify a heat map. If you're not going to do that, we don't even need lima here. We are going to use our color brewer to get colors, and that's something we'll talk about right at the start of this script. Gplots is the main package we'll use for making heat maps. It has a heat map 2 or heat map.2 function that I sort of go to for all my heat map plotting. There are certainly many other R packages for making heat maps, and you can absolutely explore them. I just find I don't need sophisticated, highly annotated heat maps with lots of text around them. I just need heat maps. So Gplots works well for me. Well, we're going to play around too with a Game of Thrones package from GitHub. So in order to install that, you would have to enter this in your R console. And you don't need to do this, by the way. It's just to show you some fun colors. And then these last 2 packages are both options for making interactive heat maps directly from the same thing we'll pass to Gplots to make static heat maps. Okay, so if you don't have these packages, and you certainly don't have these last 2, I'm guessing you don't have the Game of Thrones package, and probably don't have Gplots or R color brewer, now's the time to go over to the Packages tab, set your repositories, and start looking for those. Again, the Game of Thrones package you would install using DevTools, and it would install directly from GitHub. It's not going to be on CRAN or any of the other repos. Okay, so you could pause the video and do that now. All right. Now that we have all the packages installed and we're ready to go, I think the first thing we should probably talk about is color. After all, color is central to making any heat map. It's a visual representation of the data. And in this case, the color tells us a lot, and there's a lot of ways to mess up the relationship between the color and the data. So I guess the first thing to highlight is there are a lot of ways to assemble a palette of colors in R. And I'll give you 4 examples here of ways to do that. One way to do it is to use simple functions from the Gplots package, same package that we'll use in a little bit to make our heat map. And these functions usually involve the name of 2 colors put together. And the idea is it's going to ramp from blue to red with white in the middle, and it'll have 75 gradations on its way from true blue to true red. So I get to set the number of gradations from blue to red. I've chosen 75 here. You can choose whatever you want. But when I run this function, it's going to create a character vector of colors. And we will, I got to load the R packages here. And just as a reminder, I am running version 4 of R now. So if you see anything that looks a little bit differently than what you're seeing, it may be because of different package versions or R versions. And I had to do that, remember, in order to show you differential transcript usage in the last lecture using the isoform switch analyzer package. All right, so I'm going to run this blue-red function, and I'm going to highlight the object I made. And the first thing I see is a really key part of colors in general, but certainly how we use them in R, which is unique colors can be specified using these hex codes. Hex codes are alphanumeric, 6 alphanumeric characters together, preceded by a pound sign or hash sign, and they represent 256 colors in total. So you can really assemble complex palettes if you just know the hex numbers. Now, it's chosen the hex colors for me based on the fact that I said I wanted to start with blue, which is 4 zeros FF, and I wanted to end with red. which is FF 4 zeros. So I now have a color palette. It's that easy. But I also want you to understand there is an extensive range of color palettes already available to you, and many of those come in this R Color Brewer package. So I'm going to head back over to the slides just to highlight this. And R Color Brewer is really an R implementation of a project called Color Brewer Now 2.0, which was really developed for cartography, so making maps. And it was really to address the challenge of making colors that even when positioned close to each other, like on borders of counties or townships, would be visually distinct. And so there's a lot of design that has gone into coming up with colors here that are visually impactful, distinct, and clear. And so there are multi-hue panels, single hue panels. There are color-blind safe panels, which we'll talk about in a minute. And then these panels can be sequential, right? So same color, diverging, or qualitative. And we'll look at those in just a second. So you can access those Color Brewer palettes directly within R. We'll do that in just a minute. But here are the qualitative panels, a distinct color for each entry in the panel. Color Brewer panels have specific names, and that's how you call on them. So these are the 8 qualitative palettes available in R Color Brewer. You can see that the most number of colors you get here is 12. And this, poses a challenge if you have graphics that are going to have more than 12 colors. There's other R packages that deal with more palettes, but obviously, the human eye is going to have trouble as you start to have more colors, really distinguishing many, many colors. In addition to qualitative palettes, there are sequential palettes, and these are the names of them, and they start from light shades all the way to darker shades in the same sequence of color space. And then lastly, there are diverging palettes that start at one color and go to an opposite color on the other end with a white or light color in the middle. All right, so if we go back to R, we can see those same palettes just by running the function display brewer all. And that will generate exactly the plots I just showed you directly in your plots window. And if we want only the palettes that are colorblind friendly, we can set the option colorblind equals true and pass that option to this color brewer all function, and we get only those palettes that are colorblind friendly. Okay, so an easy way for you to pick palettes that are going to look good and play nice with different colorblind situations. All right, how else can you make colors? Well, there's all kinds of functions. The graphics devices package, which kind of loads in the background when you are doing anything in R, lets you ramp any colors together you want. You just give it a character vector of either hex codes or accepted names of colors, and it'll ramp from blue to white to red with 100 gradations from one extreme to the other. So that would be called color ramp palette function. I'm going to run that. Anytime you want to pick a palette here from Color Brewer, you use the brewer.pal function. You call out the name and the number of colors you want to pull from that palette. In this case, I want red, blue, and I want all 11 colors. There are 11 in that palette. So that gives me another palette. You could also copy and paste in your own hex codes, right? These are just creating character vectors of hex codes. So if you had your own colors that you wanted, you could just understand what those hex codes are and put them in. And there's all kinds of different apps to do that. I have one on my computer called SIP. And what SIP does is it will let you pick any color. And you can see as I mouse over, it gives me a name of the color, but it also finds that hex code. All right, I can click that and I immediately have copied that hex code and I could paste it in here. You can also save palettes like I've done here and click on any of the colors and it copies it to your clipboard and lets you paste that in as well. All right, so it should be very easy for you to use apps like this app or just searching online to find the hex codes you need. And then lastly, of course, we have our Game of Thrones package, which after you've loaded the package, it has a GOT. For those of you who are fans of the Game of Thrones TV show, at least while it was running, has the GOT function that lets you pick color spectra from any character's palette. All right, so if we go back to our presentation here, you've got a lot of palettes you can choose from, all the houses in the Game of Thrones. You can even get a White Walker's palette, a Jon Snow palette, a Lady Marjorie palette, a Daenerys, you name it. And of course, it wouldn't be complete without the option equals Arya, where you can pick sort of these Arya colors. So have fun with that. Lots of ways to choose colors in R. All right, so we'll stop there for the colors part, and in the second part of the video, we'll actually move forward with clustering our genes.